Andreas, good evening.
Good evening, Jim. Good to
Thank
see
you so much for staying up late for us.
No problem at all.
Oh, good. Well, let me introduce you and then we can begin. And as we go through the session, I'll try to pick up some questions for you.
Great.
All right, Dr. Goebel,
Great, yeah.
Associate Professor in Pain Medicine at the University of Liverpool and the Honorary Consultant in Pain Medicine at the Walton Center at the National Health Service Foundation Trust in Liverpool. He is also a member of the International Research Consortium for CRPS, and he is a avid researcher of CRPS and has published a number of articles on CRPS. Dr. Goebel will summarize the current evidence on the efficacy of immune treatments of CRPS. Welcome.
Right. Thanks very much, Jim, for the introduction, and thank you for inviting me to present. And I'm really delighted to be able to participate in this event, and I hope the whole event goes well. I understand it has only just started. Is that right, Jim?
It's wonderful. I'm going to leave you now, and I'll be watching. Take care.
Yeah. Okay. Can you hear me? Okay, Jim. Can we just confirm
Absolutely.
that?
Everything's fine.
Okay. Perfect. So I can start by saying that this presentation will not last the whole hour. I've looked at it. I'm going to go through it slowly, but even then, I think we have also time for questions if you like afterwards. And maybe we have more time for questions than what is in the program. I was asked to speak on the modulation of the immune system, and that's also the most interesting topic for me to present and update you a little bit actually where we stand at the moment. And I hope you enjoy this because a lot of things have happened, and I presume most people that are listening are patients. Now, I've seen that the conference is called Treating the Whole Person and Optimizing Wellness, and I'm not sure how exactly this will fit to optimizing wellness. I've had a few ideas, but let's see. So the immune system is not that impressive really when you look at it in terms of organs. They're all quite small organs, and actually normally the immune cells are sort of part of other organs. They're hiding in certain organs, and nevertheless the immune system is almost like is similar like a nervous system. It controls a lot of our body function. We generally think about, when we think about the immune system, we think a lot about cells. And when we look at the cells of the immune system, then they are roughly divided into the cells of the innate immune system and then the adaptive immune system. The innate immune system is that system that recognizes foreign bodies or dirt in a wound or a bacterium by its just basic characteristics, basic markers, whereas the adaptive immune system has an ability to adapt and discover, recognize new things and strengthen its own response after encountering new pathogens. It's originally, I think, at least it's still thought like that the immune system is mostly concerned with dealing with foreign bodies and pathogens. I should say at this point that I'm really fascinated about this immune system, but I'm not a immunologist. I'm an anesthetist by training. I trained in Germany and then in the United Kingdom. I now live with my family in the United Kingdom. But I had some special training in looking at the immune system, and I underwent that training in Harvard Medical School. And I went there because particularly I wanted to find a way to understand complex regional pain syndrome and other severe chronic pains. And that journey has been quite a really exciting journey, and it's been going on for quite a while, and we are discovering exciting new things along the way every year, and our understanding has developed. I hope I can share that a little bit with you. But I was just thinking a little bit about the title of this whole symposium, and is there a connection? And we should at least look at these general points. So how does the immune system matter in CRPS, and why might wellness be important? So tissue healing is often affected in people with CRPS, pointing to some immune problem. So that's clear. The immune system, though, may actually cause CRPS, and that's my main topic for this talk. Stress of having significant pain, as any significant stress, can affect the immune system. And perhaps some people with CRPS are therefore at higher risk to get simple infections such as a cold. There's no data, and we've been asked that by our patients a lot. We're a national center seeing a lot of people from across the UK. There's no data to suggest that really people with CRPS or other severe chronic pains are more susceptible to COVID. Whether that is so, therefore, I really don't know. If there is an effect, then I doubt that it is a pronounced effect. We would know that by now, and not from CRPS because that's rare, but from other similar condition, and nothing has come forward to my knowledge. But nevertheless, as we know, if we feel run down, then our immune system is affected. And then I don't know why wellness might be important. I really don't, except that taking care of ourselves and reducing overly stressful experiences is always important. So I suppose then that's where this comes in. A couple of additional words maybe that are also related to the idea of wellness and treating the whole person. Treating the whole person is part of the idea in the title of this symposium. Maybe if we have a conversation later about this, but I wonder whether CRPS can start at times when people have a particular distress, whether we are at higher risk at times when if we have an accident at a time when we also experience other distress. For example, some of my patients tell me that someone died around that time, someone close to them, or something else happened to them. But our data are actually not very good in CRPS on this, and in part that is because, as you know, this is not a very common condition, and particularly not the persistent CRPS. The ongoing CRPS is really actually rare. It's great that we have now the opportunity to communicate through large distances, but the actual numbers of people is not that high. Some of my patients that they have experienced distressing events at the time of when they had their injury and the development of CRPS. So it is possible that severe distress makes people biologically vulnerable to the development of CRPS after trauma. And I'll show you a little diagram in a little moment about this. So that it may be that the immune system gets triggered by severe distress. That is not true for all patients. Not everyone who develops CRPS had severe distress, but perhaps this is one pathway that plays a role. And we are thinking now, because we found now also in other people with severe pain, we started off with CRPS, but we found now in other people similar mechanisms as those which I'll show you now. We are wondering whether there might be a way to prevent such severe distress in the future. When we think about the immune system in CRPS or anywhere, we will generally think about inflammation. And that's really, if you think back of when I showed you the two types of immune cells, the innate immune cells and the adaptive immune cells. So inflammation is a general principle. Any foreign body that goes into our system somehow will cause an inflammatory response when we injure ourselves, when there's always a bit of an inflammation there. And it was Frank Huygens' group that first showed that there's actually a bit of inflammation also in the CRPS affected arm or leg early after the trauma. We don't know yet how important that is. And it's probably not the type of inflammation that we see in conditions that are really truly inflammatory, but it was one of the first observations that showed that something is truly not quite right with regards to the immune system in patients with CRPS, because we shouldn't have raised inflammatory markers in these affected areas. Frank Böcklein around the same time showed this in a different way. This is a neuro type of inflammation. So somehow in the CRPS affected limb, early after injury, the nerves get really activated and they secrete little mediators, and you can measure their effect. We still don't really know why they do that. So we don't know why these nerve mediators and other inflammation mediators are there in early CRPS, but they are there. So there's clearly an immune involvement. So this is all to do with the immune system. What I really would like to talk about is antibody-mediated autoimmunity, and that's a different type of immune involvement. It's an involvement where largely the adaptive immune system comes in. If you think about that branch of immune cells, that was what you saw on the right side, the T cells and the B cells are important here. And it is what we need to defend ourselves against certain sophisticated pathogens, sophisticated bacteria. We wouldn't survive a pneumonia without that system. And I'll show you now that in a bit more detail. Some of you might have seen this, and I apologize if you have. This was developed a cartoon by one of my PhD students together with me. And I'm hoping that it shows a little bit what we are after and what we and others have now found as very clear evidence of an involvement of the immune system. So normally, if you encounter a bacteria in an organism, like in our organism, if a pathogen bacteria is in our organism that shouldn't be there, then these cells, the antigen presenting cells, will capture that and will present it to the adaptive immune system. The adaptive immune system consists of T cells and B cells mainly, and some other cells. And why is that happening? It's because the antigen presenting cells, so those cells that belong to the simple innate immune system, they are not always sophisticated enough to deal with a situation where we have a lot of these bacteria. They would get overwhelmed. They need this additional system. So what happens then? The T cell gives its okay, and the B cell then shoots out these things, proteins called antibodies. And if it shoots enough of these, then they get rid of these bacteria. This wasn't basically the trigger bacteria that triggers this response, but then any other bacteria will be addressed by the B cell response. So we need the antibodies to defend ourselves against pathogens. And how do these antibodies defend us from pathogens? They do that by binding to the pathogens. So they bind to the cell surfaces, and every antibody has a different type of specificity. Every antibody is special and looks at different structures. So this antibody would not fit here, yeah, and this would not fit here. There's something in this Y-shaped area that is special and is just recognizing certain structures. So the structures that are recognized by this particular antibody are those structures to which the antibody then binds. So it recognizes the structure and it binds to it. It's a special kind of binding, so it sticks on there. And different antibodies have different ways of stickiness, but that's the main thing. It sticks on there. And when it sticks on there, then this causes then secondary, following this, it causes inflammation and the destruction of the target cell. The moment these antibodies have bound, the cell has lost, this target cell, the baddie has lost. There isn't very much they can do anymore. So the moment this is, if you like, tagged by these antibodies, then additional things happen there, which happen very quickly, and the cell will be destroyed. Now, this system is really sophisticated and it ensures our survival. But as with anything and everything in biology and in nature, it doesn't always go right. And we have conditions that are where that immune response, instead of being directed against a baddie, someone coming from outside, is directed against ourselves. And if that happens, then this is called autoantibodies. So antibodies that bind to ourselves are called autoantibodies. So here's a little self cell, that's a kidney cell. And as any cell in our body, that cell will die. We are constantly having cells dying, and after some time, we are virtually not the same anymore, because all our cells have, or most of our cells have reproduced and died, and the new cells came into their place. So if that happens, then the same process happens afterwards that happens with that I've just shown you with the foreign strange bacteria that shouldn't be there. Exactly the same. So the dead cell parts will be taken up by the antigen presenting cell, and that cell presents that to the adaptive immune system. However, there's something called tolerance, so that the adaptive immune system cannot act. It's been trained to not act. It's a complicated process, complex process, how this happens, but it won't act on if it sees that what is actually presented by that cell is actually part of our own body. Now, if we are genetically predisposed, so if we have certain genes that make us vulnerable, and sometimes even if there's some environmental damage, such as people that are much exposed, for example, to ultraviolet light. So there's some certain damage factors can break this tolerance, and that's not very good. So if the tolerance is broken, then suddenly that system again works here, and we have an autoreactive deputy here, an autoreactive B cell. Now, what do you think is happening now? Do you remember where this part came from? It came from a kidney cell. And so these autoreactive proteins will now bind to our own kidney, not the dead kidney, but the alive kidney, unfortunately. And that binding, as we've seen before, will trigger inflammation, immune cell infiltration, and ultimately a kidney disease. So that's how autoantibodies work. But we know that CRPS and other pains are not inflammatory, and they're also not really destructive in the traditional sense that we have tissue destruction. Our patients tell me that it is destructive to their mind and destructive to their lives, and I appreciate that, but it is not destructive in the sense of really destroying larger amounts of tissues. So how does that work? We think this can work in that there are some special type of autoantibodies which, although they bind to the cell, they will not trigger a destructive cascade of events. But what they do, and this is now the main and the core point, is they bind to structures and change these structures. They don't destroy them, but they change them. So here you've got these, in gray, you've got a neuron. Those are the membranes. This is the inner part of a neuron. And you've got here surface structures on that neuron that are called channels, ion channels. And you need these channels in order to regulate the life of this neuron and the electricity of this neuron. If an antibody now binds, an autoantibody binds to this, then it may change the way that these potassium channels, as they are called, these ion channels, work. So if now an action potential goes out, these ion channels don't work properly anymore. And in the case of a motor disorder, patients may develop involuntary muscle problems. And we think that that's something similar like that happens in CRPS and in other chronic pain conditions. So to summarize here, we have a neuron in red. So that is our nerve. That is more centrally and that is more peripherally in the skin. And there are several ways how these antibodies can perhaps work. They can bind to these surface structures, or perhaps in CRPS, they might not even bind directly to the neuron, as I've just shown you. So they might not work like that, but they might work by binding and changing the function of cells that are close by. And then that cell then produces other mediators that irritate that nerve, so that the nerve is constantly being irritated. That is happening in the periphery. I've seen a few questions here. Could that happen in the central nervous system? How are glial cells involved? And we don't know. So at the moment, we think that this happens in the periphery, but there are glial cells also in the periphery. So it is possible that one mechanism is that they bind to one of those peripheral glial cells, and the glial cells then produce mediators and make that neuron very sensitive. And that needs to happen for a while. So it also needs the whole central nervous system. It's not like a hammer just hitting a nail. It probably needs some time to upregulate. But then the whole electricity is jumbled up, and even a small stimulus from the outside will be painful. And the nerve probably starts to also fire independently and spontaneously. This only happens in the context of injury, as you know. So I've called this a two-hit model. Yeah, you need to have these antibodies which are produced for God knows why they're produced, but in keeping with things that happen in other people with other disorders. But here in CRPS, it's special. You also need to have an injury to the area. Somehow the injury probably changes the way these little blue things look like, so that only the injury makes it possible for these antibodies to bind. Maybe there's a few things how one can think about this, but it's a two-hit phenomenon. One needs these antibodies and the injury. So in an integrated model of autoantibody CRPS, if I can just say that again, it is possible that the brain is involved somehow initially. There is the idea that perhaps, at least for some patients, there were affecting events that affect the brain and that affect then the immune cells through ways which we don't understand. There are things called epigenetic changes. It is possible that constant distress, you need this brain in order to experience distress, that if you have that a lot, that changes the way that these B cells, so the deputies, actually work. So maybe that contributes to breaking the tolerance. You almost certainly need some genetic vulnerability, which again, we don't yet understand. This doesn't happen to everyone now, but if that is there and you have a physical trauma, then in that context, these B cells are getting activated, it appears, or they were already activated, and now they can bind to nerves that have been changed in the context of that trauma. So you need a few factors, maybe a brain factor to predispose people, likely a genetic factor. You need the trauma, and then you need these antibodies that are produced by these abnormally working B cells. And there have been, these are publications, as you can see, only over the last 2 years, and most of them are from our team and from the Stanford team with David Clark and Wade Kingery, and then with quite a lot of collaborators. So you see that they're all in quite good journals. So there's more and more coming out about this mechanism that I've just described with some variations. And I hope that I can convince you now a little bit by showing you a few results from our lab that this is actually for real. This is really happening like this. So we started doing these experiments already in 2000 and, well, actually in the early 2000s I started in Oxford. But we ended up with a model that works as follows, and anything where you use mice is called a model. We take these proteins from a patient with CRPS. The proteins are called IgG immunoglobulins. Those contain the antibodies, but also other antibodies which are not autoantibodies, which are normal antibodies. But in this mixture, there should be the autoantibodies. And we inject these antibodies to mice. Why does this work? Why could the mice be a good target? Because the mouse is not so different from us. So many structures in the mouse and in the nerves of the mouse are very similar as in humans. And this, so that one can hope, if you like, that if you do this, that the mouse can become as affected as the human. We've tried that first when I was in Oxford. I tried this and I could see that the mouse was not as happy as other mice who were injected by antibodies from healthy people. But the mouse did not develop CRPS. There was something obscure going on. The mouse wasn't quite right, but didn't develop CRPS. So we said we have to unfortunately injure the mouse hind paw under anesthesia. It's only a small injury. And you remember that two-hit model. So we inject IgG from the patient to the mouse, and we then, in the presence of the IgG in the mouse, we will then give them a small injury, and then we measure their sensitivity of their paw. That's how you measure whether someone has pain, whether a mouse has pain. You measure the sensitivity, and we call that passive transfer trauma model. And how do we measure sensitivity? I'm sorry, by putting a little, not too sharp, it's not really sharp, but a pokey little device up to the hind paw of the mouse. And if the mouse is very sensitive, you can imagine that the mouse will lift the paw even if you put just a tiny bit of pressure on there. Whereas if the mouse can't be bothered and isn't really sensitive, then it takes a long time and you increase pressure until eventually the mouse will lift the hind paw. So your mouse doesn't really have to suffer. It's just being basically asked to lift the hind paw, and with that we can measure how sensitive that hind paw is. And this is the core result really from this, and we published that last year because we managed to now show that this actually works over a longer period of time. And what you can see here, this is a normal sensitivity of a mouse hind paw. But if you then injure the mouse, as for all of us, if we have a small injury to our hand, a small cut with a knife, maybe 3 centimeters long, maybe 2 millimeters deep, that would be painful, wouldn't it? And it would be sensitive. We all have experienced that. So our hand would remain sensitive, wouldn't it? But it would also recover quite soon. And so what you can see here is the recovery of the mice that were injected not with CRPS IgG, but with healthy control IgG. Whereas here you can see the non-recovery of the mice that are injected with CRPS IgG, and that difference is highly significant, and it stays here. So as long as you make sure that there's still human IgG in these mice, even so the injury heals, it completely heals. There's no more, the paw looks absolutely normal by that time, but something has happened, and these antibodies somehow bind somewhere. We are not sure where they bind, and render these mice sensitive. And you can see in the other paw that has not been injured, nothing happens. Nothing happens. I hope that makes sense. And then as a final, if you like, scientific slide, I think the others are not like that. If you in that system then give the mice that are so sensitive a drug, an immune drug, and here we gave them anakinra, then you can, if you're lucky, reverse that process. So if you can test out, you can use this model for testing drugs and see whether certain drugs might work. And we've done that, and that was published in a very respected journal, and we have subsequently managed to get some money, and we are trying that drug out now. I'm not saying that this is going to work. I'm not saying that we can immediately transfer everything from that model to the human, but it's clear that, and this is a traditional and classical way of showing this, a very solid way of showing this, there's got to be an autoimmune reaction at least involved in causing persistent CRPS. So in people that have it 7, 8, 9, 10 years, that's where we took these antibody from patients who had it that long. So we think there's still an ongoing active immune process. It's not all in the brain. So here just once more to highlight, this little line shows the group of mice that was here still very sensitive, injected with CRPS IgG. And here they still have the CRPS IgG, but they are also treated with a certain drug, and that drug reverses the effect of these antibodies. So I've been asked to outline what that means in terms of immune treatments. And I can see that people are asking questions and so on, but I'm unable to see that at the same time or respond to it. So I'm hoping that Jim will pick some of these questions up, and then we can discuss these later. So what does this mean if these antibodies are involved in CRPS? There's quite a lot of interventions that can now be considered. And they can be classed into 3 categories. Firstly, interventions that can reduce the antibody concentration in a patient's blood. And again, here there are 3 ways of what that can be done. Firstly, they can be physically removed. We can just physically, with a filter, remove these antibodies. And that's been done in some centers in the US. And we've tried this, and we were the first to actually publish on this. This is from a US center, where you can see that if the pain initially was high, then quite a number of people after this cleaning event, if you like, reported afterwards having lower pain levels. And as a result of our laboratory and clinical studies, this is now possible. CRPS is part of these conditions that could be considered, are now considered reasonably to be considered for this cleaning process, which is called plasma exchange or plasmapheresis. So we've had a number of patient groups, and we learned from patients that they think it's a okay thing to do, even so there is some trauma involved, because in order to be able to do this, one needs to put quite a significantly big needle in one extremity of one blood vessel in order to get enough blood out. So the blood is taken out, is then cleaned, and is then returned. In my experience here in Liverpool, there are truly patients who got better. It has so far almost in everyone needed repeating after a while. And there are some side effects because the trauma of the needle can, in some patients, cause special types of pain at the place where the needle is inserted. So I'm skeptical how far we can use this, but I think we've shown the principle that this could work. It hasn't been shown in the best way possible. It hasn't been shown in a randomized trial. Now, another way to go about reducing these antibody levels in the blood is by disabling or destroying the cells that produce the antibodies. You remember the deputy. The deputy is a B cell, and there are now for rheumatology and neurology actually quite a number of drugs developed already that were developed specially to affect these cells, because you can imagine this CRPS is not the only condition that is caused or contributed to by these antibodies. There are others. And so there are now a number of drugs available. So, and the drugs again can be classified in some ways, and one of the simplest and earliest drugs is disease-modifying drugs, and we've tried that out, and I thought we had actually some success with that. But probably by just giving this drug, that's not enough. But still, I think we saw a signal that that could work. There are just recently a few drugs that additionally come out, and there are more and more coming out. Then there's another way to do this, and that is you can reduce the antibody levels pharmacologically. You don't destroy the B cells, you don't remove the antibodies physically, but pharmacologically, and high-dose IVIG can do that, and there's a few new drugs out that can do this. As you know, we tried low-dose IVIG. We thought we had a response, and probably some people can respond to this. But it did not work in a large trial, so it's only a very few people. But the high-dose IVIG has not been tried. So this was the first way of how we can take this forward. Another way is to look at downstream effects. So once the antibodies have bound, do they maybe cause a little bit of inflammation around the area, as you've seen in this mouse model before? So we thought that we can interrupt the mouse model by a drug that affects the effects of the antibodies. So it doesn't change the antibodies, but it changes what then happens to the cell after the antibodies have bound. And then finally, and that is future music, if you like, if we come to understand what exactly the target is, if we come to understand exactly what is happening here, what structure exactly do these antibodies bind to, and we are currently trying to find that out, and some other people are as well. It's not easy. Then we can maybe create a drug that is custom designed, if you like, to fit in here and interfere with that interaction between antibody and part of the cell. And I mean, this is now part of the baddie cell, but what we want is interfering with the interaction between the antibody and part of the goodie cell. Maybe I should change that picture a bit to not confuse people. And then finally, there could be combination of all of these. So that's pretty much what I wanted to say, except that this also has an implication for diagnostic tests and biomarkers. So given that we are on a good way to show that there is a special thing in people's blood which shouldn't be there, then we should be able to develop tests that use that presence and just tell people, yeah, you do have CRPS. This is how these tests could look like. We don't have a CRPS test, but it's a similar principle, and it's been done with many, many other conditions. And I think we are on a good way on developing that. And why is that important? It validates your condition. It is clear. Often doctors still don't believe in chronic pain because it's nothing you can measure easily. What we showed you what I've shown you in the mouse model is just possible so far in the lab. It's better than what we had before, but it's not yet possible in a hospital. So if we have a simple test, you can go to the doctor and they can do a blood test. And I think we are on the way to do that. Diagnosis will also be faster. And I think we can also use these then to monitor treatment. So that's another benefit of this research. So I thought I should, at the end now, just say a few words about this and the context in which this happens. So I have just now found something similar with collaborators in London in a different pain condition, which is much more common than CRPS. And you wouldn't believe how much bigger the interest is by industry and funders and others in our results now. So I think for us to push CRPS and push new solutions for CRPS, we need a patient voice that is becoming political. And that has happened in some other conditions, but it hasn't really happened very much in CRPS. So one way for patients to become involved is to talk to politicians and so on. They are doing that in part here in the UK, but not enough. And I think the United States, as being such a big country and also a relatively rich country, even if it might not feel like that, would be at a unique place to engage in a process where patients push the politicians to make funding available. We are now at the state where from my perspective, we could now try drugs, for example. There are existing drugs which you could try, but to do one of those trials, for one drug, you got to spend about $600,000. It's nowadays not possible in any other way if you really want to have a result. That's what everyone spends. And the question is, can we shout loud enough to say, in CRPS, we need to do that? And I think we need a patient voice there. And it's really you, people that listen to this, who I'd like to encourage and entice to think, could I do something? Could I speak to people? We have, I think, for the first time now, enough evidence to justify, only since the last year, I'd say, to justify trying suitable immune drugs. But we need funds to do this. And it doesn't have to be us in Liverpool. It can also be other people. But whoever does this, these funds don't come from nowhere. We need these funds somehow. And as an example, we did get a donation by a US philanthropist, which was in excess of $500,000, which has permitted for us to now set up a small study in anakinra to just try whether this might work. And we know that both we and others can do more. So I think there are pathways, and probably you are much better able to engage with these pathways. We are competing, if we are just looking at government grant-giving bodies, we are competing with people that do children's eye diseases to people that do cancer. It's a huge competitive field. As you know, we have won competitions a good number of times, but still, in order to make progress, I'd like to encourage you to get involved. So think about how you can support RSDSA and researchers in the field. Do you know of people who can help? Can you develop ideas how to do this? Okay, that's pretty much what I want to say, and I look forward to listen to your questions and maybe have a little discussion about these things. Thank you.
Andreas, before we begin questions, two things. First, I wanted to thank you, and secondly, we're funding David Clark's study that we're really hoping that he'll find some biomarkers at Stanford. And the second thing, during November, which is CRPS Awareness Month in the United States, we're going to be working with our advocacy committee to develop a short postcard for everyone to really contact their federal House and Senate representatives to really talk about CRPS as a rare disease and that it is the most painful disease there is. And if you're not treated well, you're sentenced to disability, perhaps. So we're on that way.
Yeah, I'm glad to hear that, and that's brilliant. But I'd say, you know, let's not trust that this is just being done for us. I mean, there's maybe 100 people listening to this, or 150. My experience is really we need everyone to think about this, and RSDSA is a great organization to sort of channel this. But we probably need everyone listening to think about how you might be involved. The help often comes from areas where you really don't expect it. I've experienced that so many times. But yeah, thank you.
Okay. Well, one of our earliest questions was looking at a question regarding Lyme, and could you talk about a possible association with Lyme and their sporadic reports of antibodies being effective?
Yeah, I mean, you know, the idea is that this autoimmune response or immune response might have been triggered by encounter of a pathogen, and that's how some autoimmune disorders can start. And so it's not out of the question that could be triggered by Lyme, but we don't have the evidence that we would at this point be able to consider as conclusive evidence for that at all. So there's very little out that. So I'm agnostic. I can't really say very much about this.
Okay. We have another question. Is there a link between the gene MC1R and CRPS? And she further says, I have red hair, blue eyes from Scottish ancestors, and I've noticed that many people with CRPS in Canada also have red hair and red beards.
That's great. You should publish that. I don't know. Maybe we should look into this. We've just actually done a genetic study with people in Cambridge, and I'll ask them that. I'm not aware of anything, but you know, that's an interesting observation. It does seem so that so our next symposium, and COVID has helped us in a way with that, will very likely be online and next year, and we will have speakers from around the world, not just from the traditional countries. And the CRPS does really exist also in India and in sub-Saharan Africa. But yeah, maybe that is an additional risk factor. I don't know.
There also was a question about mitochondria and how CRP affects it, or vice versa.
Yeah, I've seen this. For us now, we are looking also at the mitochondrial genetic information. I am not aware that we have yet a conclusive study that has looked at that. I think so far there's nothing conclusive out there. I think it's, yeah, people are looking at this in a number of pain conditions now, and perhaps something will come out of that. It's not my special area, I'm sorry. I doubt that this is a mitochondrial disease in the sense that everything can be explained by mitochondria, but maybe they are a risk factor.
Okay, thank you. There were a number of questions about supplements to boost immunity. Any thoughts?
Supplements,
vitamins, medical foods.
Yeah. I don't know. When we say boost immunity, then so there's quite a lot of stuff out to boost immunity so that you get less colds, or might be less susceptible to the flu. And I'm not an expert on that. I'm really sorry. I'm just an expert on a relatively small area. So that may well be that this could help CRPS. There's really nothing published that we can take serious so far. Once there is something published, I'd be super happy to tell everyone about it. But that it can help CRPS itself is at the moment not established in any way.
Okay. There was another question about, does inflammation show up in the segmentation rate?
Yes, it should. In the so there's something called ESR, and that is usually raised in inflammatory disorders, and CRPS is not an inflammatory disorder. So that's one of those special things about it. So if there's inflammation, it would be a very localized, very confined inflammation, for example, just around nerves, which does not reflect in the ESR, in the sedimentation rate. So no, it won't come out there. So this is what is special. You can't detect it in this way.
Okay. Anyone else have any other questions I might have missed? If you could type them now. There was a question. There's been a lot of interest, and we also funded a study at Stanford on LDN, low-dose naltrexone, and it seems to have a strong effect on glia activation. Could you talk a little bit about that, please?
Yeah, I mean, this drug is meant to have an immune property, and I know you're funding a study on this, and I haven't seen the results of it yet. So that would be, if you like, it would be in that class where you have a general immune modulation effect, if that works, and maybe it is working, or it is not, I don't know. So we haven't seen the results of the study. It's really always very pragmatic. You have to wait until you see the results. And that's all I can say about it from what I know so far.
Okay, and you quite definitively, right, a second ago said that if I understood you correctly, that CRPS is not an autoimmune disease. Is it a subset? How do you characterize it?
Say that again, please, Jim. Say that again.
I thought you said that CRPS is not an autoimmune disease. Is there a subset of people with it have that autoimmune component?
No, I didn't say that. What I said is it's not a generalized inflammatory disease. Okay. So, for example, rheumatoid arthritis is what we call an inflammatory disorder, and it's like when you have an inflammation that is so strong that you can actually take some blood, you can do this sedimentation rate, and there's a few other things, and you can see that there's inflammation in the body, and you can't do that with CRPS. So that's the definition of what we call an inflammatory disease. But there may be microinflammation somewhere. I think in terms of autoimmunity, all bloods that we've tested from people with persistent CRPS, so longer than a year, they all had these antibodies. And so I think that generally it's probably, from our research so far, I think it's generally probably an autoimmune disorder, but one that's probably a lot different from known autoimmune disorders. And as you know, the Stanford team have now also found different type of autoimmunity, very likely being involved in early CRPS. So that's not my special area, because also clinically, I only see generally the people that have CRPS already for half a year or longer, not the very early group, but now the Stanford people have found that. So I think it's probably a major role in CRPS.
Okay. There was a question about, can CRPS cause low in uniglobin E?
I think not. We have not seen that when we tried it. It probably can do it in single cases, but we have not found anything when we checked that showed that this group, IgE, that's called, is generally reduced in patients with CRPS.
Okay. Dr. Tarfik at Stanford just published a pilot study on hydroxychloroquine as treating CRPS. Do you have any comments on that?
Yeah, I've seen that, and I mean, it's great that other people go and look at that, and it's at that stage where she now also needs to basically go and apply for funds and see whether it's worthwhile to develop, properly show that. So it's not it's a very, very, very preliminary study. but which is encouraging so far, and which would you always need then additional studies to look in more detail.
Okay.
And also to really confirm that this is real, because things can happen if you just give a patient a drug that are related to chance or to other factors. So you can't necessarily say from a type of study which was published there whether this effect is real, and that's the same for us. So we've done things like that. What's quite good is if you can show that this doesn't work. If it doesn't work in an open study like that, then you can pretty much say you don't have to go any further, because CRPS is not associated with a very a large placebo response. So if 10 or 15, we have to think about how many, but if you have maybe 15, 20, 25 people who do not respond to a certain treatment, then even if it's given openly, then you can say, well, okay, so there's no point to continue on.
Okay. Dr. Goebel, will you share your email with individuals? Would you welcome correspondence with our community?
I absolutely cannot respond to single request. I mean, I get a lot of emails from the US from people that ask whether, what they can do, and it's I try to respond to those, but I have really no capacity to engage in that very much. So of course, my email, everyone can see my email. It's quite available, but please be mindful that I might not be able to respond very quickly or respond at all. But I'm happy to share the PowerPoint. I mean, I think you've recorded that anyway. But if someone wants to have the PowerPoint as a file, basically, I'm happy to send it to you guys and then for that to be shared.
Okay. Well, I want to thank you for staying up so late for us. And if people don't know, it's 5 hours difference in the UK. And he was just such a gentleman and scientist to come help us. I want to thank you, and hopefully we'll be together at an in-person meeting in the future.
Yeah, okay, so have a great meeting, and I do apologize for not being able to answer all the questions. I see there are other questions, but just not able to. But really have a great meeting, and stay well and stay safe. Take care.
Again.
Bye.